After studying this module, you shall be able to understand what are single nucleotide polymorphisms, in short SNPs, and how they are detected, and what is the forensic application of SNPs. Single nucleotide polymorphisms, in short we call SNPs, are single base pair positions in genomic DNA at which different sequence alternatives or alleles exist in normal individuals in some populations, wherein the least frequent allele has an abundance of 1% or more. SNPs are found in human genome about once in every 1000 base pair. Given that the human genome is 3.2 billion base pair long, we can estimate that there will be approximately 1 million differences between two genomes that are due to SNPs. This represents approximately 85% of human genetic variation. The vast majority of polymorphism intrinsically limits the information that can be gained from the analysis of any given SNP, and this has been the major factor limiting their application to forensic analysis. Between 50 and 80 SNPs are required to achieve the same level of discrimination as current STR based methods. However, the vast number of SNPs within the genome, which are currently over 10 million SNPs, have been placed in public databases, can compensate for limited information carried by any individual SNP and make them a tempting polymorphism to exploit for forensic application. The technology that is being used for SNP detection is evolving, and SNP analysis is becoming possible in many forensic laboratories. Now, as regard detection of SNPs, there are many techniques available for resolution of SNPs. It was well established that particular enzymes produced by bacteria can be used to cut the DNA molecule by recognizing specific sequences. Restriction digestion can be used to genotype SNPs when the SNP either creates or destroys a particular restriction enzyme recognition sequence. But the method is limited for forensic casework because it needs a large amount of DNA and is a long and laborious process. The figure here shows that SNPs are created when the DNA replication enzymes make a mistake as they copy the cell's DNA during meiosis. The enzyme incorporates the wrong nucleotide approximately once every 9 to 10 million bases. In the vast majority of cases, SNPs are biallelic and only have two different alleles. In the alleles shown above, the thymine nucleotide has been replaced by a cytosine. Sanger sequencing, also known as chain termination sequencing, was developed in late 1970s and is a milestone in the development of molecular biology. The sequencing takes advantage of the biochemistry of DNA replication. The first stage of the analysis is to amplify the target region using PCR. Amplified products are then used as the template. in a sequencing reaction. The DNA sequencing reaction is similar to PCR amplification, and the reaction mixture is very similar, containing the thermophilic talc, DNA polymerase and deoxynucleotide triphosphates. It differs from PCR in that only one primer is used, and in addition to the DNTPs, there are 4 fluorescently labeled dideoxyribonucleotides. Each dideoxyribonucleotides is labeled with a different color dye. The DD and TPs do not contain the hydroxyl group on the 3 carbon, which prevents any extension of the DNA molecule. The concentration of DNTPs is higher than the DD and TPs, and therefore, in most cases, a DNTP is added. The DDNTPs are incorporated at random intervals along the molecule. This produces a range of different sized molecules. The products of the sequencing reaction are analyzed using capillary electrophoresis system such as ABI prism genetic analyzer that separates DNA to single base pair resolution and can simultaneously detect 4 different fluorescent labels. Sequencing is not a practical option for analysis of SNPs in a forensic context. Most SNPs are widely dispersed throughout the genome, and a separate reaction is required for each SNP. An exception is the mitochondrial genome, where a number of SNPs are concentrated into a small area and can be analyzed in a small number of reactions. Sequencing has also been a powerful method to type SNPs within rapidly evolving regions of DNA in HIV virus. This figure shows where a primer anneals to the template strand. This is extended by talc polymerase until the DDNTPs are incorporated at random, which leads to a collection of extension molecule that differ from each other by 1 nucleotide, as shown above, labeled 1 to 8. The 4 DDNTPs are labeled with different fluorescent dyes that are detected using capillary electrophoresis. Restriction digestion and sequence analysis are not viable method to use for forensic cases that might require the analysis of 50 to 80 SNPs dispersed around the genome. A number of methods have evolved that can be applied for detection of SNPs. The primer extension. Primer extension is a robust method for discriminating between different alleles, and several methodologies have been developed. One of the commonly used method is mini sequencing reaction. The basis of the reaction is very similar to Sanger sequencing. The first part of the procedure is to amplify the target region using PCR. An internal primer then anneals to the denatured PCR product. The 3' end of the primer is adjacent to the polymorphic site. The primer is then extended by talc polymerase, but only DDNTPs that are labeled with fluorescent dyes are provided. The primer is only extended by 1 nucleotide. The extended primer can be analyzed using capillary gel electrophoresis, and color of the detected peak allows the SNPs to be characterized. A widely used commercial kit called snapshot provided by applied biosystem is based on this methodology. By using different size primer and different fluorescent tags for each of the 4 bases, a large number of SNPs can be simultaneously detected. Variations on the primer extension technique include pyrosequencing, microarrays, where the extension primers are attached to a silicon chip, and allele specific extension, when the primer is only extended if it is 100% complementary to the target sequence. The figure shows the steps of extension. Here the primer extension assays, the target sequence is amplified using PCR and the products are used as the template in the extension assay. Number 2, an internal primer hybridizes to target adjacent to the SNP, and a single fluorescently labeled ddNTP is added by Taq polymerase, and in the third step the reaction is analyzed by capillary electrophoresis. Allele specific hybridization. Under stringent condition, even one nucleotide mismatching between a template and primer can differentiate between 2 alleles. There is a large number of methods that exploit hybridization of probes, including reverse dot plots, Taqman assays, light cycler assays, molecular beacons, and gene chips. Now, coming to forensic application of SNPs, a vast amount of data is available on different SNPs in human genome, and one of the biggest tasks when applying SNPs to forensic application is to select the most appropriate SNPs from the overwhelming number that are available. The choice of SNPs is very much dependent on the application. For forensic identification, vast majority of forensic DNA analysis involves the characterization of biological material recovered from the scene of crime. Several panels of SNPs have been developed that are designed to provide maximum discrimination power for forensic identification. These contain SNPs that are polymorphic in all major population groups. A panel containing 52 SNPs is used for identification, which produce match probability ranging from 5 into 10 raised to power minus 19 in Asian population. However, even with the high discrimination power, the efforts involved in analyzing 50 SNPs is greater than when undertaking standard STR analysis. The major attraction of using SNPs with current technology is that SNP analysis can provide results from highly degraded DNA when conventional STR profiling has failed. SNPs are also used for prediction of geographical ancestry. In many cases, the identification of the population group from which a crime scene sample has come can be valuable information for investigating agencies to know whether the person who left the material at the scene of crime was Caucasian, Asian, African, or of mixed ancestry. Panels consisting of mitochondrial DNA, SNPs, and Y-SNPs are useful in providing information on either the maternal or paternal ancestry. Autosomal SNPs that have different frequencies in different major population groups can provide valuable information on geographic ancestry. Many of the SNPs selected for this purpose are associated with coding regions that have been subjected to selection pressures. These includes pigmentation genes, genes involved with metabolism of xenobiotic. The pigmentation gene, in addition to providing information on geographical ancestry, can also give information of phenotype of the person who deposited the biological material at the crime scene, including skin, hair and eye color. SNPs, when compared to STR loci, we find that the advantage of STR over SNPs is their power of discrimination due to large number of alleles they have in comparison with biallelic SNPs. In contrast to STRs, around 4 times more SNPs are required to reach the discrimination power equivalent to STR loci. Another major disadvantage of using SNPs is that mixtures of 2 or more people might be either problematic or impossible to interpret, since SNPs are biallelic markers. Also, current DNA databases consist of profiles comprising STR loci, and therefore SNPs cannot be used in that context. At the same time, it is possible to analyze hundreds of SNP loci, and due to their structure, the amplicon size can be much smaller, typically less than 100 base pair. This allows analysis of DNA template which are highly degraded and may generate information when standard STR typing fails to generate results. The table showing comparison between STR and SNPs would give you the idea how SNPs are better in degraded sample and how STRs are better as far as the discrimination power is concerned. In future, STRs are likely to be the most commonly used genetic polymorphism analyzed in forensic science. They are tried and tested in most judicial systems and also form the basis of most forensic DNA databases. The use of SNPs to specialized application, for example, SNP based blood grouping, profiling of badly degraded sample and molecular autopsy, looking for mutation that can explain sudden death, is likely to become more widespread. In summary, we can say that SNPs are single base pair positions in genomic DNA at which different sequence alternatives or alleles exist in normal individual in some populations wherein the least frequent allele has an abundance of 1% or more. SNPs are found in human genome about once in every 1000 base pair. Sanger sequencing, also known as chain termination sequencing, was developed in late 1970s and is a milestone in development of molecular biology. The sequencing takes advantage of the biochemistry of DNA replication. The first stage of the analysis is to amplify the target region using PCR. Amplified products are then used as the template in sequencing reaction. The primer extension is a robust method for discriminating between different alleles. The basis of the reaction is very similar to Sanger sequencing. The first part of the procedure is to amplify the target region using PCR. An internal primer then anneals to the denatured PCR product and 3' end of the primer is adjacent to the polymorphic site. Under stringent condition, even one nucleotide mismatching between the template and the primer can differentiate between two alleles. Several methods that exploit the hybridization probes, including reverse dot plots, Tachman assays, light cycler assays, molecular beacons, and gene chips are available. The great advantage of STR over SNPs is their power of discrimination due to the large number of alleles they have in comparison with biallelic SNPs. Thank you.